Bom dia a todos. A aula magna da pós-graduação do Instituto de Ciências Biológicas da UFMG é um evento que marca o início do ano letivo. Agradecemos a presença de todos que estão aqui. Daremos início à cerimônia de abertura da aula magna da pós-graduação do ICB 2023. Para a cerimônia de abertura desse evento, convidamos então o diretor do ICB, o professor Ricardo Gonçalves, a coordenadora do Núcleo de Apoio à Pós-Graduação do ICB, a professora Érica Cristina Jorge, e a subcoordenadora do Núcleo de Apoio à Pós-Graduação do ICB, a professora Glória Regina Franco. Agora, para falar em nome do Instituto de Ciências Biológicas, seu diretor, o professor Ricardo Gonçalves. Bom dia, professor.
Bom dia a todos. Estou te ouvindo muito bem. Queria agradecer o convite, né? Eu acho que esse é um momento muito especial para nós, no início do semestre. Agradeço, então, a professora Érica, a quem eu já cumprimento, né? Então, meu bom dia, professora querida Glória, Amanda, todos os participantes aqui. E esse é um momento importante para a gente de reinício, né? Quase que diante de uma normalidade, depois desses anos de pandemia. A pandemia ainda não acabou, mas a gente vive um pouquinho mais na normalidade, graças à ciência, graças à vacina, graças às medidas que deveriam ter sido tomadas, né? Talvez até mais precocemente, mas a gente fica feliz de estar podendo aqui. Infelizmente, ainda nessa transmissão online, seria excelente ter o Dr. Krause aqui para nós ao vivo, né? Presencialmente. A gente vai trabalhar isso, viu, Eric e Glória, com todo o apoio do ICB, tá? Eu acho que a gente vive um momento muito especial depois dessa pandemia e que todos nós fomos afetados, né, pessoalmente, nos nossos trabalhos e principalmente as nossas pós-graduações. A gente tem vivido aí um grande desafio da retomada desse trabalho, né? Eu acho que a gente, todos nós aí vinculados a programas de pós-graduação, e a gente tem essa noção porque o ICB tem 16 programas de pós-graduação, a gente tem visto que esse ataque à ciência que nós vivemos nos últimos anos e a falta de recursos e de apoio nos trouxe um cenário desafiador, né? Sem dúvida nenhuma, a gente vê que a procura dos alunos pela pós-graduação tem diminuído bastante. A gente tem, então, aí um desafio pela frente que deve ser enfrentado e eu tenho certeza que a gente vai conseguir, né, retomar todo esse sucesso e apoio que são necessários e são inevitáveis. Não há como a gente sobreviver, né, sem o apoio à ciência, e o Brasil tem uma história gigantesca, e o ICB, que é um dos maiores institutos de pesquisa, ensino e extensão da América Latina, tem um papel fundamental nisso. Nós temos aqui muitos INCTs, muitos coordenadores de rede que são coordenadas na FAPEMIG aqui no ICB, e eu acho que essa tradição nossa toda vai ser fundamental para que a gente retome esse sucesso e essa busca, né, importante pela ciência. Tá? Então, eu agradeço, eu acho que a gente está abrindo, né, sem dúvida nenhuma, eu acho que essa aula magna com chave de ouro, né, eu acho que é uma apresentação interessantíssima, importantíssima, e eu acho, né, e eu parabenizo aí o NAPG por ter conseguido, né, esse contato tão importante. Eu acho que vai ser enriquecedor e mais legal ainda e mais importante que isso fique gravado no YouTube para que quem não puder ter vindo aqui hoje, ter o prazer dessa apresentação ao vivo, que possa, então, ser presenteado com ela depois, né, na eternidade das mídias sociais, né, e assistir isso, porque eu tenho certeza que vai ser realmente um sucesso. Agradeço aí a organização, agradeço o convite, né, é sempre um prazer participar de vocês. O NAPG é um sucesso para nós, então agradeço a Rose, querida, que é sempre muito competente nessas organizações também, e que seja bem-vindo o Dr. Krause aí com a sua apresentação. Um abraço a todos.
Obrigada, professor. Agora vamos passar a palavra para a professora Érica Cristina Jorge, que é a coordenadora do Núcleo de Apoio à Pós-graduação. Bom dia, professor.
Bom dia, Amanda. Primeiramente, eu gostaria de cumprimentar o professor Ricardo Gonçalves, diretor do Instituto de Ciências Biológicas, Também gostaria de cumprimentar a professora Glória, subcoordenadora, parceira no NAPG. Cumprimento também os demais presentes, professores, discentes e todos aí presentes na nossa sala virtual. Muito bom dia. É com uma imensa alegria e satisfação que realizamos hoje a aula magna da pós-graduação. É um evento que dá início às atividades do ano letivo de 2023. Gostaria de agradecer, em especial, a presença da equipe do NAPG, que organizou esse evento com muito carinho e dedicação. A nossa equipe é composta, além da professora Glória e eu, né, pela nossa técnica administrativa, querida Rose Batista Castro, e também pelos discentes Amanda Moreira, Bruna Oliveira, Flávia Santiago, Júlia Meirelles, Vivianícia e Wesley Ferreira, que são discentes de pós-graduação dos 16 PPGs sediados no ICB. Você, discente que está nos ouvindo hoje, se tiver interesse em participar da equipe do NAPG, por favor, entre em contato, que a gente vai adorar ter a sua ajuda nos nossos eventos. Deixo aqui o meu muito obrigada de coração pelo trabalho incansável dessa equipe. Antes de darmos início à aula magna, eu gostaria de desejar que a gente tenha um ano letivo de muito trabalho e muito sucesso, sem deixar de lado o respeito, a generosidade e a boa vontade uns com os outros. Aproveitem essa aula magna, que vai ser sensacional, tenho certeza, assim como todos os outros eventos que estamos planejando para a comunidade do ICB em 2023. Fiquem atentos aos eventos do NAPG. Muito obrigada a todos e uma boa palestra.
A professora Glória Regina Franco, que é a subcoordenadora de pós-graduação do ICB. Bom dia, professora.
Bom dia, Amanda. Gostaria de cumprimentar inicialmente meu colega e meu amigo, professor Ricardo Gonçalves, diretor do ICB, a minha queridíssima parceira, a professora Érica Jorge, também as pessoas que estão nos bastidores dessa transmissão, Rosemary Castro, Flávia, a Bruna, e também muito agradecer imensamente ao professor Johannes Krause, que prontamente aceitou o nosso convite, e principalmente ao professor Fabrício Santos, que foi o intermediário aqui nas conversas com o professor Johannes Krause, né? O professor Fabrício é amigo do professor Johannes e o convidou em nome do NAPG e fez todos os contatos, né, com o professor Johannes. Também agradecer ao discente Tomás Pinotti, que é aluno de doutorado do programa de Genética, orientado pelo professor Fabrício Santos, que vai estar aqui, então, apresentando para vocês o professor Johannes Krause. Então, eu gostaria de reforçar o que disse a Érica e desejar a todos um excelente ano. Essa aula magna é muito especial para o NAPG, porque a gente abre todas as atividades da pós-graduação do ano, né? É um evento anual, e a gente tem sempre um convidado muito importante, e a escolha do professor Johannes Krause foi devido ao que aconteceu no ano passado na premiação do Nobel, que foi dado ao professor Svante Paabo, que trabalha na área de paleogenômica e DNAs ancestrais, e o professor Johannes Krause participa desse trabalho também. Ele está no grupo do Svante Paabo, então ele tem muitas coisas interessantes para falar para a gente aqui. Certamente será uma palestra maravilhosa, então foi muito especial ele ter aceitado o nosso convite nesse momento, né, em que a gente vai celebrar esse prêmio Nobel aí importante na área de genética, de antropologia. E eu agradeço também a presença de todos que estão aí no YouTube assistindo essa aula magna, e eu espero que seja tudo excelente. Também gostaria de falar que vocês podem fazer as perguntas, porque nós temos membros da NAPG que vão transmitir as perguntas do YouTube aqui para a nossa sala de transmissão. As perguntas podem ser feitas em português, e aí a gente traduz aqui para o professor Johannes, que não fala português, claro, né? Mas fiquem à vontade de fazer suas perguntas, e aí nós transmitiremos para o professor Johannes. E desejo a todos, então, um bom semestre e uma boa aula magna. Muito obrigada.
Obrigada. Só alguns avisos para todos de confirmarem a presença nesse evento através do link que está na descrição do vídeo e do sorteio de brindes para os presentes. Assim, é imprescindível que vocês façam o registro da presença. As perguntas, como a professora falou, podem ser feitas, podem ser enviadas pelo chat ao longo da palestra. E agora, então, nós convidamos o senhor Tomás Pinotti, do Departamento de Genética, Ecologia e Evolução, para apresentar o convidado de honra, o professor Johannes Krause, que irá proferir a palestra da Aula Magna de 2023. Bom dia, Tomás.
Bom dia. Oi, gente, eu vou falar em inglês porque aí eu já faço o link com o Johannes, ok? Hello, everyone. My name is Tomás, a PhD student at the Department of Genética, and today I have the pleasure and honor to introduce one of the leading figures in everything ancient DNA related, Professor Johannes Krause. Thank you so much, Professor Johannes, for accepting our invitation. Before I give you the word, I'll try to make sure everyone is so psyched as I am to hear your talk, and I'll tell them how cool your research is. Professor Johannes Krause is director of the Department of Archaeogenetics of the Max Planck Institute for Evolutionary Anthropology, located in Leipzig, Germany. He has pioneered many directions of the highly diverse field of ancient DNA, and he has done research on topics as distinct as ancient DNA to document the spread of Indo-European languages and the origins of Yersinia pestis, the etiological agent of the Black Death. He has very recently published the genome of Ludwig van Beethoven from ancient locks of hair from the German composer, and with a group from Universidade de São Paulo, he has also investigated the genomes of 10,000-year-old individuals from Lagoa Santa, related to the famous Luzia, of course. If this was not enough, he has also had an active participation in the first analysis of not one, but two ancient extinct hominins, the Neanderthals and the Denisovans, the latter one we didn't even know existed before the work of Johannes' team. Thank you so much, Johannes. The stage is yours.
Thank you, Thomas. I guess the slides go up now. Perfect. Yeah, thank you all. Thank you for joining. I'm very sad to not be with you today for not just the reason that it's always much better to meet people in person, but also because this is how the Institute currently looks like. So we have still winter here, surprisingly late this year. So I would very much enjoy, obviously, to be with you in Brazil right now. So I have titled the presentation today, Deep History of Humanity, because I want to really spend the whole focus of our work that Thomas just nicely summarized in a few sentences. So kind of going all the way from Neanderthals to the Black Death and the origin of the Black Death, and kind of the topic base is archaeogenetics, which is the field that I'm working in, combining archaeology and genetic research. So also to some extent spanning the bridge between the humanities and the natural sciences. So much of our research has only become possible over the last few years because the technology, how we study DNA, has changed a lot. So there was a bit of a revolution on the prices and on the throughput of the machines that decode DNA. So this is just an overview of how much genome was costing in like 20 years ago, in 2001 or so. For one genome, you actually paid several hundred million euros. And then because of the machines that got better and better, and the throughput of the machines that got better and better, you can see that the costs have dropped dramatically. So 15 years ago, we had this type of machine in our lab, and that could produce about 100 DNA sequences per day. And today we have such machines in our lab, they produce 20 billion DNA sequences per day. So the throughput of the machines has increased by 100 million fold. And therefore, we can, of course, produce data 100 million fold faster and, of course, more. And that has really caused this kind of revolution in our field, not just in archaeogenetics, but in genetics in general. But what we are using the DNA for is some sort of time machine, because we're using DNA from the past to learn something about genetic diversity of past population or past species. And DNA has the wonderful property that it's a very stable molecule. It survives for a very long time. The oldest genome that has been decoded so far is more than 1 million years old from a mammoth from Alaska. So it does preserve well, because evolution has made sure that our building blocks are stable, because otherwise if they break, it's a mutation, so that is bad. So therefore DNA is a stable molecule that survives thousands of years under at least certain conditions. So what we do is we take ancient skeletons, can be human remains, can be animal remains, and we extract the DNA in so-called clean rooms, where we try to avoid to contaminate the samples with our own DNA, where there's of course a high risk, because such a bone has very little DNA preserved after such a long time. We drill in little holes, we release the DNA in some sort of extraction buffer, and then the DNA gets prepared for high throughput DNA sequencing. There are some steps often in between that then allow us to then reconstruct at the end on the computer from many tiny little DNA fragments the genome of people or organisms from the past. And for humans, we have done that for 10,000s of human remains from all over the world. So this is just the data that we have produced in our lab over the last 5 years or so. So more than 20,000 ancient skeletons we have analyzed from more than 100,000 years of human history, from more than 2,500 archaeological sites. And you can really see we are working almost everywhere in the world, trying to reconstruct genetic history. So trying to reconstruct the genetic diversity of people from the past to learn how they came where they are today and how they changed through time. And that also tells us something about the evolution of humans and how we evolved over the last few 100,000 years. So combining genetic data that we now have with also the data of archaeologists and anthropologists, we have pretty good understanding now about how humans evolved and how they spread over the planet. So we evolved in Africa some 200 to 300,000 years ago as a species, modern humans, Homo sapiens. But then about 40 to 50,000 years ago, humans moved out of Africa and came to Europe and Asia. And then later on, about 15,000 years to 20,000 years ago, they also moved into the Americas, settled then also North and South America. And when they came to Europe and Asia, there were actually already people there, but different types of people. So Neanderthals in Europe and other types of hominins in Asia. So in East Asia, Homo erectus was potentially still alive, but that's not uncontested. So some people question whether Homo erectus was still around when humans came out of Africa 50,000 years ago. But there was this other type of human that we call Denisovans, which is kind of an Asian type of Neanderthal that split from the Neanderthal lineage about 300,000 years ago, and that we discovered actually in the lab about 13 years ago, when we analyzed a little finger bone and actually found out that that finger bone is not a Neanderthal, it's not a modern human, but it's something new that we could then call Denisovan. But there are also other types of human forms, probably about 4 or 5 species or different distinct forms of humans were around when humans left Africa 50,000 years ago. So for example, in Indonesia, on the island of Flores, there was this Homo floresiensis, which is very tiny, small hominin that had a quite small head and small brain. It was discovered in 2003 when Lord of the Rings was in the cinema. It was small people with big feet, so they are called hobbits. So sometimes you actually read about them and they're referred to as hobbits just because of their stature. And one of the big questions we had then in the past was, what happened when humans, modern humans, Homo sapiens, came out of Africa and met those other forms of humans? Did they actually interact with them? Did they exchange genes? Did they have offspring? Did they have children? Did they have sex with each other, basically, or not? And when we first had the Neanderthal genome then, in 2010, we could ask that question and see, do people that live in the world today carry Neanderthal DNA? So did Neanderthals and modern humans have offspring with each other? And we did find that this is actually the case. So in all people outside Africa, so Europeans, Asians, Australians, Americans, Native Americans, we do find about 2% Neanderthal DNA, which we have interpreted with a common admixture event that happened sometime when humans left Africa, maybe about 50,000 years ago, and they went through the Middle East. And the Middle East had Neanderthals at that time. And from there, this population went to Europe, Asia, Australia, and the Americas later on. But they took this Neanderthal admixture that occurred than 40 to 50,000 years ago into all those other parts of the world. And this is why we see it now all over the world outside Africa, but not in Africa, because Neanderthals were never present in Africa. We then asked the same question with the Denisovans. The Denisovans actually genetically contribute to people that live in the world today. And there we also found that some populations in the world today carry Denisovan DNA. And that is East Asians, so people that would live in China today, they have about 0.2% of Denisovan DNA. But populations in Papua New Guinea, in the highlands of Papua New Guinea, so the Aborigine population there, as well as Aborigines from Australia, they have about 5% Denisovan DNA. So they not only have 2% Neanderthal, they also have 5% Denisovan. So there are some populations that have received more of this type of archaic human ancestry, how we sometimes call it, so those ancient hominins, than others. So in Africa, we don't find that at all. But then outside Africa, we have Neanderthal, and in Southeast Asia, also this Denisovan DNA. One big question that then my PhD advisor, Svante Paabo, had was, what can we actually learn from that type of DNA about what makes us human? So Svante, who you've just heard, received the Nobel Prize just a few months ago, and probably you've heard that before. He had the great idea to then use the genomes of those people from the past to then learn what is so special about us, modern humans. Because when we have the genome of a chimpanzee and when we have the genome of a Neanderthal, we can then compare those genomes and then find positions in the genomes where chimpanzees look like Neanderthals, but modern humans are different. Because those will be positions that changed in our lineage after Neanderthals and humans diverged. So those are genes or positions in the genome that are human specific. Those are mutations that happened in our recent evolution that are not shared with Neanderthals and chimpanzees. And this is really important because those are then the genetic changes that made modern humans modern humans. And why is that important? Because, of course, modern humans are somehow special because we are the last hominin on the planet. All other hominins, so Neanderthals, Denisovans, Homo erectus, Homo floresiensis, all those other hominins have gotten extinct by now. And we're the only surviving hominin lineage, but we're also super successful. We have colonized the entire world within a few thousand years. We have developed technologies. We have developed the Neolithic package, so a new way of food production that wasn't around before. So people were only hunter-gatherers before modern humans. So we are very, very successful, very smart. Obviously, that's kind of the secret of our success. We're 8 billion people now on the planet. And there should be a biological basis for that. So it's something that made us so smart, so successful, so also in some ways aggressive and so mobile, and also adventurous to some degree, because also humans have crossed water or land bridges, for example, coming to the Americas 15,000 years ago that no other hominin did before. And with this tool, ancient DNA, by extracting DNA from ancient fossils, this is now possible. We now have a catalog, thanks to Svante's work, of the things that make us human and where we are genetically different from, say, Neanderthals or our closest living relatives, such as chimpanzees or other great apes. What we as archaeogeneticists are also quite interested in is genetic diversity. And the genetic diversity can be informative, not just about evolution, so how phenotypic traits change. Like, for example, you look at those 4 people here and they look phenotypically different. So you might wonder what happened to them and their ancestors and how do those phenotypes change. But we can also use the genetic differences that we see between individuals and populations to tell human history, to tell the history of the population. So how did the population, for example, end up where they are today? Where did they come from? What changed through time? How did certain people move through time and space? And that is something that we call genetic history. So we don't necessarily need a written record about a certain event, but we can actually use DNA to tell us about things that happened in the past. And one way how this is often done these days is by then doing something that we call a principal component analysis. So that's something you see on the left side. This is an analysis that was already done more than 15 years ago by a colleague of mine, John November, where he compared different individuals and their genomes from different parts of Europe. And what you have here in this colorful cloud are all individuals. And what this is the principal component analysis, that they analyze the genomes of those people. And what you see here is the genetic distance between the individuals that are plotted on this map. There are about 3,000 people from different parts of Europe that have their ancestors, their grandparents from the same region. And after the genetic distance was calculated, they were colored by where their ancestors, where their grandparents were coming from. And everyone who said my ancestors come from Spain got this bluish color. Everybody who said my ancestors come from Portugal got purple. And this is France, Switzerland, Belgium, Great Britain, Ireland, Scotland, Netherlands, Norway, Denmark, Germany, Sweden, Austria, and so forth. What you actually see here on this map is the map of Europe that is written into our genes, which was a big surprise to a lot of people, because it basically means that the topography of some part in the world is written into the genes of the people that live there today. And this is something that we call isolation by distance. So it's just when people move into a certain space, they're genetically changed, there's genetic drift, so they're slowly changing through time. And then people that live close to each other, they're genetically closer than people that live far away from each other. So it's a higher probability to have children with someone that lives in your vicinity, close by, than someone that lives far away. And through time, that basically the topography of the region then writes basically into our genome. Because if there is something like, you can see that between Spain and France, there is this empty space here. This is the Pyrenees. This is a mountain range. Then here below Switzerland and Italy, you have again an empty space. That is the Alps. Then here you have again an empty space. That is the Adriatic Sea. And then down here you have the Mediterranean Sea. And here you have actually the Atlantic. So basically you really have those spaces, the geographic spaces, that you then also see in genetic space. And one big question that people then had immediately when this type of analysis were done is, was it always like that? Or did that change through time? So did it always look like this? Or if we go back in time, were there major changes because people moved around, because people migrated from A to B? And this is basically what we're doing now. We're reconstructing genetic structure, like you see here on the left, through time to see if there are changes, because those changes might be indicative of a movement of people, of a change, because people might have moved from A to B. And there's many different time periods that you can look at and study that type of genetic history. And this is just an overview for Central Europe here, where you have different time slices, different time periods that are often called based on the culture, like Paleolithic and Mesolithic and Neolithic, or the Stone Ages, and then Bronze, Iron Age, the Metal Ages, Antiquity, and so forth. Most of that is outside historical documents. Most of our history, nobody wrote anything about. So therefore, we know very little about those things that happened. And then you only have archaeology, and the archaeology might be telling you something, but often not for sure if, for example, people have moved. Because you might see that there's a change in the archaeology, but that might not necessarily mean that it was new people coming from somewhere. It could simply mean that it is a cultural development. So people just come up with new ideas, and therefore with new technologies, and then slowly, basically, the archaeology is changing. And one of the biggest events that we observe in many parts, almost in all parts of the world, in different time periods though, that is a big change in our archaeology or culture that we find with the people from the past, is the so-called Neolithic Revolution, which happens in Central Europe about 7,500 years ago. It happens in the Near East about 10,000 years ago, in China about 10,000 years ago, in Southeast Asia about 4,000 years ago, in the Americas, depending where you are, between 5,000 and 3,000 years ago. It's in different parts of the world at different times, but it does happen everywhere, which also tells you about that all humans basically are capable of that type of large scale cultural change. But what people don't know and did not know is whether this change in culture, in so-called subsistence, so from hunting and gathering to an agricultural lifestyle where people suddenly have domestic animals and where they have fields and crops, so domestic plants, whether this change is that just new ideas that people came up with, or was it actually people that moved around and brought new technologies to other places? So a question that we often refer to as pots or people. So if you see a change in culture, is it just ideas, or is it actually people introducing those ideas? And this is something that only based on archaeology is a bit hard to measure. But if you have genetic data, you can actually directly test that. So if it's just culture that changes, you would have such a scenario here where you have, before the Neolithic, the same genetic makeup of the people than after the Neolithic. So just the neighboring population might tell the people about the new ideas, but there's no people moving. So the genetic landscape that we've seen would be the same, wouldn't change. But then there's the opposite, and unfortunately it's not animated slides right now, but you would see it here. If it's endemic diffusion, you would actually see that there is a genetic change, that suddenly this green component is present after this Neolithic population came in contact and then probably moved into the space of the people before agriculture. So you will see a genetic shift, a genetic change. And this is something that we have been looking at over the last 10 years or so with some of the first studies that we published about 2013, 14, 15. And I want to summarize some of them here, where we collected the first few hundred genomes from the past. So you see here the different ages. So the oldest ones here are about 9,000 years old. The youngest ones are about this BCE here, about 2,000 years old. And basically what we have now is a time transect from Europe where we can see through time how the genetic landscape changed. And first we start this little time travel now with about 8,000 years ago, where we look at the people in Europe that lived here 8,000 years ago. Those are all hunter-gatherers. This is before agriculture has been introduced to Europe. And you can see in the background here, those gray dots are the people that live in Europe and the Near East today. So this would be Western Europe, Central Europe, Eastern Europe. This is the Balkan. This is Anatolia. This is the Caucasus. This is the Near East. This is Northern Africa. You can see there's a large gap here. This is the Mediterranean. There is a gap here. This is basically the Black Sea. So it's basically the European map, but a bit kind of tilted to the side. And those dots that you have over here, those are people from the past, from 8,000 years ago, the hunter-gatherers. They're clearly falling outside the genetic makeup of the people that live in Europe today. So they're distinct. That's kind of a first piece of information here. What we have here now are Anatolian farmers. So the first farmers, because farming, at least in this part of the world, emerged in the Near East, so in the southern part of Anatolia, they already had farming at 8,000 years ago, and they're genetically very different to the, say, people that lived in Central Europe at the time, which we would call Western European hunter-gatherers. So there's a big genetic shift and change between Anatolia at that time and European hunter-gatherers at that time. And now we move in time forward from 8,000 years ago to about 7,500 years ago. And then we suddenly have this group of people, but they are this time not from Anatolia, they are actually from Central Europe. So from this region here, so from the Central European region, and they're genetically very different to the people that lived in Central Europe before, the hunter-gatherers, but they're almost indistinguishable from the people that lived in Anatolia around 8,000 years ago. So we really have very good evidence there was a major migration, a major genetic shift, a shift in ancestry, and therefore a movement of people from Anatolia to Central Europe between 8,000 and 7,000 years ago. This is then the agriculturalists, so those people in their archaeology, they're associated with farming. So farming was clearly brought to Europe from Anatolia. And that is even still reflected in the people today. So more than half of the genes, half of the ancestry of people that live in Europe today comes from Anatolia, from that movement, from the early farmers to Europe. But it's also clear that more must be going on, because this is how Central Europe looks like today, and this is how it looked like in the past. So the story isn't over yet at 7,000 years. If we move to about 5,000 years, which is the so-called Chalcolithic, the Copper Age, you can actually see that the individuals move again towards here the left, and they are actually moving into the direction of hunter-gatherers, and indeed in their genomes we suddenly have chunks of hunter-gatherer DNA, which tells us that the farmers did not replace the hunter-gatherers, but over time they actually mixed with each other, so that by about 5,000 years ago all of Europe looks like the green triangles down here. There is still some people, you can actually see them in the background, there's some gray dots that look like the Chalcolithic people. And those are the people that today live in Sardinia, on this island in the Mediterranean. And this is interesting because it seems to have preserved the type of genetic makeup that was in all of Europe about 5,000 years ago. But then again, something more must happen because 5,000 years ago, people in Central Europe look like this, and today they look like that. So more kind of must happen. And here we now have a population from the Bronze Age. So those are people that lived also about 5,000 years ago. They already had bronze, but they're now from Eastern Europe, where this is called the Bronze Age. In Europe, we still call it the Stone Age, so it's sometimes a bit confusing. But those people are genetically very different to the people that lived during the early Bronze Age or the Copper Age in Central Europe. So there's a big genetic difference between Eastern Europe and Western Europe at that time. But then about 4,800 years ago, so only 200 years after this time period, this all comes together. So we suddenly seem to have another movement of people, so a movement of people from Eastern Europe into Central Europe, because Central Europe suddenly is those, for example, orange squares here, they are from Central Europe. So from here down the Chalcolithic time, they move to up here, and some of them are even indistinguishable again from Bronze Age people from just 200 years earlier from Eastern Europe. So again, there's a major movement of people from the steppe, from the Eastern European steppe into Central Europe. And those people here that move in have a different lifestyle. They're actually pastoralists, so they're herders. They're very mobile. They have just invented wheel and wagon, and they are really mobile, moving, as it seems, quite fast towards the east, because they also arrive in the Altai Mountains in Siberia, but also into the west, so coming to Europe. And within a few hundred years, they're found everywhere in Europe. This is just an overview of the data that we had about last year, 2022. So we have many more dots than you see on this map, but they all confirm the same story. We have those basically three major genetic components coming into Europe: the hunter-gatherer component, the one from the Anatolia with early farming, and then the what we sometimes call Yamnaya or steppe component that comes in later, that forms then the green dots that you see here in the middle, which is modern day in the back, and then Bronze Age people here at the front, which also tells us that from the Bronze Age onwards, basically not major genetic changes happen. So in the last 3,500 years, it has been more or less quite genetically stable Europe without major incoming populations. So only within Europe we have changes, but not so much from the outside. So in summary, what can we say here? We have this early Neolithic migration that started about 8,000 years ago, introducing farming to then Central Europe from Anatolia. Between 7,000 and 5,000 years ago, you have admixture between hunter-gatherers and modern humans. And then about 4,800 years ago, you have the second larger migration that happens then from Eastern Europe to Central Europe, those people with wheels, wagons, mobile people, maybe horses, even though that's contested right now. They come in and actually replace largely the population that lived here before. This actually, the second migration that happens is largely male-driven. So we see 7 times more male than female individuals moving in. So there's all kind of speculation why that happens. We're not quite sure. Could have many reasons. Some people say it was conflicts in warfare. Some people say it was disease. Some people say it was just a lifestyle, that herding was superior to farming at that time. We will maybe never find that out, or maybe it's a mixture of all 3 things together. Well, Thomas already said at the beginning, we cannot just then use this type of information to tell us something about the change of people, but potentially also the change of languages. Because we do have, of course, a correlation between people moving and the languages they speak moving. And especially for the languages that are mostly spoken in Europe today, the Indo-European languages, there's 2 main hypotheses where they came from. One is the so-called steppe hypothesis, stating that they came from Eastern Europe during basically this time period around 5,000 years ago with this Yamnaya expansion. And the other one is the Anatolian hypothesis, that they came with early farming about 8,000 to 9,000 years ago to Europe. And of course, I've just shown you there were 2 big migrations, one from Anatolia, one from Eastern Europe. So in a way, genetically, you might say, can you really distinguish between the two hypotheses? But we can, because they also make a prediction what type of genetic material you should find in India, because the languages connect northern India and western Eurasia. But in India, in fact, especially in northern India, we do not find Anatolian ancestry. We actually have no evidence so far of a major movement of people from Anatolia, early farming moving from Anatolia to India. But we do have, starting about 3,600 years ago, a major influx of steppe ancestry from what we found basically in Eastern Europe into northern India. And that correlates almost perfectly in India, the amount of steppe ancestry, with being an Indo-European speaker or a Dravidian speaker, which is the other larger language family that is present in India today. So we think that based on the genetic data, we have much more support for the steppe hypothesis that Indo-European languages came to Europe from Eastern Europe about 5,000 to 6,000 years ago than the Anatolian hypothesis, which also is, of course, important because if that is true, then the languages spoken in Southern Europe, like Basque, for example, which is a non-Indo-European language, are leftover languages from the Anatolian languages. So it's actually an early farmer language, that the Basque language that is still present in Iberia today. And you have things like Paleosardo, Etruscan, or Minoan, which are also non-Indo-European languages and probably were also languages of the early farmers that came from Anatolia about 8,000 years ago. What we can also look at is the phenotype through time, because we have, of course, genomic data and the genome codes for the building blocks. So we can see how things have changed through time. And there's many different phenotypes we can look at. Here's just a few examples, like eye color, for example. There's a variant that gives people light eyes, like blue or green, which is here in red. And you see that the frequency today in Central Europeans is actually quite high. It's a recessive gene, so if you have only one copy, you still have brown eyes. If you have two copies, you have blue eyes, for example. And you see if you kind of look the time back, around 6,000 years ago, the frequency was much lower. So the time when people came from Anatolia, brought farming here, probably most people had dark eyes, so brown eyes. But if you go back to the hunter-gatherer time, before farming was introduced, those people are fixed for actually light eyes. So all hunter-gatherers that were living in Western Eurasia around 8,000 years ago had blue eyes, as it seems to us right now, based on our data. We can also look at skin color, where we see actually quite an opposite pattern. So the skin color, which is so typical for Europeans today, if you look at the frequency today, it's fixed. Like all Europeans basically have two genes that give them light skin color. But if you go back in time, you can actually see that the frequency goes down. So the early farmers had a lower frequency of one of the variants and intermediate frequency of the other one. So they probably had darker skin in average than the people that live in Europe today. But if you go further back in time to the hunter-gatherers, you can actually see one is 0 and another one is present in Eastern Europe, but not in Western Europe. So in Western Europe, it seems that all hunter-gatherers before 7,000 years ago, so before agriculture was introduced, had dark skin. So their phenotype was like that. So this is actually the phenotype of Europeans 8,000 years ago. It's very different to how a lot of people might think about Europeans 8,000 years ago. So they really got light in their skin color only in the last 8,000 years. Only when agriculture was introduced, people became light-skinned. And there's a probable reason for that, because when you have agriculture as a subsistence, you don't have a lot of vitamin D in your diet. And when you move to the north, you cannot produce vitamin D from sunshine in your skin, which is one way to produce it. You have to get it through your diet. And if your diet doesn't have a lot of vitamin D because you're not eating a lot of fish and meat, one way to adapt is get light skin. And that's probably what happened in Europe. And that's probably when people changed to agriculture, they became light-skinned, because that's another way to produce vitamin D in your skin if you're living up in the north. As Thomas also already introduced you, we've been also working on the genetic history of the Americas. And based on the genomic data, the genetic data we have so far, and I know that there's some archaeological data that kind of questions some of the genetic data, but based on the genetic data we have so far, the Americas were settled roughly about 15,000 years ago and after. So about 15,000 years ago, people start from Alaska moving into North America. And based on our genetic data we currently have is that there's two main lineages. We call ancestral A and ancestral B. Ancestral B is present in Native American populations today, and in the past only North America. But then there's this other stream, this ancestral A kind of genetic lineage that splits up and moves pretty early on, probably around 14,500 years old already, because that's when we have archaeology, like in Chile, for example, into the Americas, into South America. And then there's actually two streams. There's an earlier one, the green one here, there's a blue one, and then there's even a third one. So there's three different types of ancestral genetic lineages that spread into South America. The first one about 10 to 12,000 years ago, then another one about 9,000 years ago, and then this yellow one about 4,500 years ago, probably introducing also some of the agriculture, so some of the Neolithic package into the Andes, so probably related also to the domestication, for example, of maize, so corn, as well as the domestication of potato, for example, and then the basically exchange between North and South America. And this is quite interesting because, as Thomas already mentioned, we have, for example, then analyzed individuals like Lapa do Santo, for example, that have this really early ancestry, whereas some of those other individuals here have rather this type of green ancestry. So we have basically these two different types of ancestry present in the Americas early on, and that homogenize only about 5,000 years ago. So then they're really kind of homogeneous in the genetic makeup. And then from about 5,000 years ago in the Andes, for example, which we analyzed in a follow-up study, we can then even see that the genetic structure of the people is pretty much the same like it is today in South America. So from about 5,000 years ago, like we've seen in Europe from about 3,500 years ago, the Bronze Age, it's very similar in South America, that basically not bigger things happen until, of course, in the Americas with the colonial age, with this massive genetic influx from Europe, Asia, and Africa. But that basically until about then 500 years ago, the Americas were actually quite stable. So you have very similar genetic makeup of the people 500 years ago and 5,000 years ago in most parts of the Americas. And this is just the data set that we had generated for that. So about 50 genomes we have analyzed from many different parts of the Americas. That gives us this type of information, and then really allows us to have a time transect and see basically the same thing that I just showed you for Europe, also for the Americas, those genetic shifts and changes through time. In our work, we are, however, not just interested in the DNA of the people and what we can learn about the evolution and the history of the people, but we're also interested in the pathogens, so the bacteria and the viruses that were present in the people in the past that might have killed them. And that's a type of research that I pioneered then some 12 years ago or so, where we then started to analyze pathogens from the past by reconstructing entire genomes of ancient bacteria and viruses from skeletons that we analyzed. So not just looking at the human DNA, but also looking at the pathogen DNA that might be present in those people. And we did many different organisms. So we've looked a lot into Yersinia pestis, and I will say a few words about that in a minute. We've also studied Mycobacterium leprae, the causative agent of leprosy, but we've also looked into syphilis, so Treponema pallidum, and its origin probably in the New World. It's probably one of the few diseases that has been introduced from the New World to the Old World. We also looked at Mycobacterium tuberculosis and its evolution, especially in the Americas. We're really fascinated by studying ancient Mycobacterium tuberculosis from Peru, from Ecuador, and from Colombia. And what we found, which is still very puzzling and surprising, is that Native Americans before contact carried tuberculosis that today we only find in seals and in sea lions. So it was probably a zoonosis from seals that were present on the western coast of South America that were taken up into the human population and then basically passed on between humans. So it was a zoonotic origin of tuberculosis. Only later on in the colonial age, they got European tuberculosis, which is the dominant tuberculosis that you find in the Americas today. We also looked at Salmonella enterica, so typhoid fever, where we found that some of the large epidemics that happened in the Native American population after contact were actually caused by typhoid fever. So we had the introduction of this disease by the Spaniards probably already in the 16th century, causing some of the large-scale epidemics, for example, Mesoamerica and Mexico, like the Coccolizli, for example, that seems to have been linked to Salmonella enterica. And then we looked at the evolution of this disease over the last 8,000 years. And we also looked at Helicobacter pylori, which are bacteria that live in the stomach of people, where we had the fortunate luck that we could work on this fellow here, which is a reconstruction of the so-called Iceman. So the mummy that was stuck in the ice for about 5,000 years. It was found in the early 90s, and we could study the frozen stomach and reconstruct Helicobacter pylori from that frozen stomach. Just a few words about plague, because that's kind of where you've seen we have done most of our research. There are three big pandemics in plague. So one is the Justinianic plague that marks the transition between antiquity and the medieval time. So big pandemic killing millions of people. The Black Death is the second pandemic, probably the biggest killer in human history, killing half of all Europeans in the mid of the 14th century. And the third pandemic, which happened in East Asia mostly, but also then brought the disease to all over the world, including actually South America. So this introduction happened in the late 19th century by steamships and rats on ships and into the harbors, and from there then into the country. And that is then the third pandemic that then stopped basically only in the antibiotic age, when we had very powerful treatments against Yersinia pestis. So the first study that we did more than 10 years ago was on the Black Death, because back then it was actually not known what pathogen has caused the Black Death. And we were able to reconstruct from a cemetery in London complete genomes of Yersinia pestis from that time period, because this cemetery had only been used during the Black Death. So we are rather sure that this indeed was Black Death that we've seen there. We could then compare the Black Death from the past to modern day Yersinia pestis diversity, and what we could see is that the London strain falls basal to the vast majority of strains that are circulating in the world today, but it does fall on this branch 1 here. So those are different Yersinia pestis strains that are circulating in rodent populations all over the world, and you can see this is actually the common ancestor on this branch 1. And one of the questions that we then had over the last few years is, where did it originate? Where did it actually come from? It caused the Black Death. But where did it come from? Did it come from Eastern Europe, Central Asia, East Asia, Northeastern Asia? There were all kind of hypotheses, as you can see here, of people, where did it actually originate? And we were able to find one interesting site here that we worked on with Phil Slavin, who is a historian, and he studied the site called Karadijah, which is close to Bishkek, the capital of Kyrgyzstan. And this is a very particularly interesting site, because at the site you have gravestones with writing on them in Syriac, and it gives you the name of the person, gives you the age, so when the person died, and it gives you the reason why the person died of, and it actually said that those people died of pestilence in the site. And this is 8 years before the Black Death. So we thought that could be the place of origin. And what Phil Slavin also found was that there is an overrepresentation of people from 1338 on the cemetery. So this is the years, this is the number of gravestones, and you can see there's a lot of them in 1338. So we analyzed the ones from 1338 to see whether maybe they have plague. And indeed, Maria here at the lab, she found that they had Yersinia pestis. We could reconstruct an entire genome from the site Karadijah. Where did it fall in the phylogeny? That actually fall right at the most recent common ancestor of 80% of all the circulating Yersinia pestis strains in the world. And it's literally the common ancestor. It doesn't have a single, what we call derived position, so something that only the genome from that time has. So it is really the mother of all the strains that have basically diverged here. So in a way, it's a bit like patient zero, or if you want, we're all experts now in pandemic. It's a bit like Wuhan, right? It's like finding Wuhan in the past. We have found the place where this strain has originated and then diverged into the different strains, like then the one that caused the Black Death in Europe just a few years later. And this is also supported by animal research. So there is reservoirs, so rodent populations, they that carry Yersinia pestis in the region. Actually, the closest related rodents are living in the vicinity of the site. So this is again Karadijah, this is Kyrgyzstan. You see all those rodents here carrying a type of Yersinia pestis that is very closely related to the one that then gave rise to this diversity. So the one that then basically emerged just before the Black Death in this region. So we really think we have basically found the region where this strain originated. So by that, I hope I could give you a little bit of a taste what archaeogenetics is about, how it informs us about genetic history, how it informs about human evolution, how it also informs us about the evolution of pathogens and disease, maybe even how they adapted through time to become human pathogens, how they changed, how we adapted to them. So there's a lot of interesting information that we can learn by studying the past, and now not just by looking at archaeology or looking at skeletons, but also looking at the DNA, the building blocks through time, which of course allows us directly in situ to study evolution and not just basically inferring evolution by looking at modern genetic diversity like we might have present in certain parts of the world today. So by that, of course, I want to thank a lot of people. So those are the people that worked with us on the Settlement of the Americas project, so including Johannes Krause, who is in Sao Paulo, and Lars Feldschmidt and Cosimo Post and David Reich. Also the rest of the department, we're quite a diverse group, as you can see, a lot of funding agencies. Also want to make you aware that I've written a kind of popular book. Some of you might have seen it. It's also available now in Portuguese, or Brazilian, I should call it, because I don't think it's actually available in Portugal, but it is available in Brazil. So if you want to read it, I think it's a bit more kind of a layman version of what I presented today, but I think quite accessible. I wrote it with a good friend of mine who's a journalist, so hope it's actually approachable, but it's also available in many other languages. And by that, I thank you for your attention, and I'm happy to take a few questions if we still have time for that.
Well, I have to say that I am very impressed. Thank you very much for your talk. It was marvelous. It was very, very informative, Johannes. Well, we have questions in our chat that came from the audience in YouTube. Maybe we can put in the screen, and Thomas can ask to you.
I should ask, or the question. Well, yes. So I think the question has 3 parts, even though it's the same question, right? And so the question is, many paleoanthropologists in the recent years, after the publication of the hybridization between Neanderthals and modern humans, have suggested coming back with the subspecific terms Homo sapiens sapiens and Homo sapiens neanderthalensis. And I wonder, what's your paleogenomic view on that?
I've always been extremely careful, and I think this is because I'm from Svante school, to use any Latin in any of my writings. I think you will not find Latin in any of my papers, and there's quite a few. It's a very strong statement to say, you know, they could hybridize, they could not hybridize. The thing is, the species concept by Meyer is not the only one. So the idea about having fertile offspring is what defines a species. And based on Meyer's definition, Neanderthals are the same species as modern humans. However, I once gave a student the task to come up with other species concepts, and he found another 25 concepts. And based on some of them, there will be 2 species. On some of them, they will not. Good example is a coyote. I say a coyote is not a wolf, but they can have fertile offspring. A polar bear and a brown bear, they are certainly not the same species. I mean, just look at them, right? I mean, they're highly specialized Arctic animal, and then there's kind of, you know, everywhere found bear, but they can have fertile offspring. So should that be the same species? No, they're not. So, and I think that's true for many other species. And it's hard for us to do that in biology to start with. How should we do it for paleontology? Where it's even harder because you don't really know even most of their behavior. So I think we should get away from that. I think it's an academic exercise, right? And put a label on it. It's Latin. It needs to be this or that. But nothing in biology is discrete, right? Everything is a gradient. Species are also a gradient. At some point, they cannot hybridize anymore, and then more and more they kind of move away from each other. And they sometimes still have gene flow. They were never really like polar bears and brown bears because they overlap with the Pleistocene and the habitats kind of moving, and they will not diverge ever completely. So I think it's just something that it's just an academic exercise. I don't want to, I think it doesn't make a lot of sense. We can now measure based on the genomes how similar two species are, or populations, or forms, or whatever you want to call them. And I think that's much more informative than, if they are, 3 Latin words or 2 Latin words. I think, subspecies. It's anyway, because it also brings us in, how do we do that then in humans? Should we then also have subspecies in humans? Then we come almost to this kind of race question. Same thing there, humans don't have subspecies, right? I mean, humans is a gradient that runs all over the world. And I think, now that we have all those tools like genomics and genetics, we can describe animals much, much better than ever before. We can decode the entire genome, right? We can have exactly the number of positions, how they're different or the same. And I think that's much more information than 3 or 2 Latin words.
Strong statement. Yeah, I have a comment on that. We have to reanalyze the concept of species as we are reanalyzing the concept of genes. And then that's very, 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 very important. I really liked your interpretation. Yeah, species cannot be seen as names, just names, and that's very, very interesting. Yeah. Oh, go ahead, Tomás. Yeah, just a comment.
Can I make a short question before we pass to the next one, because it's kind of related? So as we're talking about Neanderthals, I'm really curious on the Denisovans, because if I have the story correct, it was the pinky bone that you're analyzing as part of your PhD, is that correct? And then you had that eureka moment that you just go out in the lab one day and then suddenly you leave the lab knowing that there's different extinct hominin species. Can you tell a little bit how that happened? I'm really curious to hear.
No, that was certainly a eureka moment, and certainly the kind of biggest, discovery I made in the lab in my career, because you just look at the genome or DNA information and you make a first tree, and then it's like, wow, this is not a Neanderthal. This is something very different. In fact, when we discovered it, we thought it's even more different than at the end it turned out to be, because we only had the mitochondrial DNA. And the mitochondrial DNA as well, in fact, as we now know, the Y chromosome of Denisovans is very, very different to Neanderthals and modern humans. So modern humans and Neanderthals are closer related on the mitochondrial DNA and the Y chromosome. But the nuclear DNA is a different story, which in itself was really puzzling for many years. So nuclear DNA, Neanderthals are closer to Denisovans, and modern humans are an outgroup, which we now have actually, because of also many other analysis, have a pretty better understanding that there was actually gene flow between early humans and Neanderthals, between Europe and Africa around 300,000 years ago, which didn't go to Asia, which only Europeans received. And the mitochondrial DNA and the Y chromosome of Neanderthals today is actually gene flow from Africa. So actually modern human DNA, which is fascinating. But it's actually confirmed because now we have Neanderthals from 400,000 years ago, before this gene flow occurred. They have Denisovan Y chromosome, and they have Denisovan mitochondrial DNA. So we actually knew that because of that, this gene flow happened. So this is very exciting. But yeah, it was an amazing moment. I mean, I remember when I was calling, Svante was my supervisor at the time, and he said, you have to sit down. I found a new human in the lab. I didn't find it in the cave. I found it in the lab. And he was amazed, and then we kind of came back and we analyzed and we looked at the data together. And I think I had this moment where basically I asked everyone in the lab at that afternoon to come to the seminar area, which is in fact outside the door because I'm back at that same institute, and I said, like, show me I'm wrong. Kind of destroy what I found, because I can't believe it, and nobody could. So until now, so it's an amazing discovery. It's a great moment, certainly, to discover a new human. And then it made me even more proud in a way than when Svante got the Nobel Prize last year, because that was the Neanderthal genome and the Denisovan, and the Neanderthal genome was my PhD project, and this one was my postdoc project. So I'd say that was pretty awesome to then see, that this is kind of some of the reasons. And obviously he's kind of done many more works and kind of followed this work after 2010 when I stopped doing this and kind of did other things. But I think it's feels also some sort of support for that as well.
I can imagine. Okay, so we have plenty of questions. I'm hoping you have booked the whole afternoon for us, Johannes. So the next question is, what about the school morphology change in the Americas around 8,000 years, the Paleo-American hypothesis and those different, like the ancestral and derived morphology? Do you see any genetic correlation when you look at the data?
We don't, and this was one of the big surprises. We did not really see a major correlation between, say, the ancestral A and the kind of lineage that we have in Lapo di Santo, so that have that kind of other, kind of more archaic, if you want, or different skull morphology. So we didn't really see that, and it's still puzzling to us why that isn't the case, but it would probably argue for that this is just some sort of morphological development which is not related to any kind of larger population movement. It's something we also have in Europe. I mean, Upper Paleolithic, the first people that came to Europe morphologically are very different to the people that lived here 15,000 years ago. Genetically, they are continuous. So there's no big thing happening between 40,000 years ago and 15,000 years ago in Europe, but the morphology of the people actually changes quite a lot. They are much more robust 40,000 years ago than they are 15,000 years ago. And I think the same might have happened in the Americas. And there's maybe just a trend. It's an overall trend that we observe in modern humans for the last 300,000 years. They become more gracile. So this is just something that happens everywhere. Why that happens, we do not know. There's other funny things happening. People become blue-eyed and kind of green-eyed in Europe. Why is that happening? There's no biology. There must be something about the kind of sexual selection or something like that. It's really hard to explain. But yeah, we don't see a correlation right now, at least.
That's very interesting. Thank you. Okay, so the next question is about pathogens, and it's naturally about the origin of the pathogens. I think a lot of the questions are about this, because we know that it's very difficult actually to establish which pathogen came, from where exactly, when this movement from the Americas and from Europe. And the question is, how do we know that syphilis was a Old World to New World movement?
Yes, New World to Old World movements, or the other way around. So how we know that, without giving you too much information about our unpublished data, we have, first of all, in both treponemal species, the major species that we have in the world today, a diversification event that is around 1500. So the moment that the Old World and the New World come in contact, the diversity increases a lot in all treponemes. In Europe, we have a pandemic. The first records we have, historical records we have in Europe, are from 1494. There were about, I think it was 20 or 30 ships that had crossed by that time between North America and Europe. After that, basically, syphilis was, spreading all over Europe. Within the next decades, it actually caused this large pandemic, you could call it syphilis pandemic in the 16th century in Europe. And it really seems to be strongly correlated to the moment that the New World and the Old World meet. So therefore, there's an old historical hypothesis that it was introduced from the New World, just based on historical evidence. In terms of the paleopathological evidence, it wasn't as clear. There was some evidence for some sort of treponeme in Europe before. We have genetic data now from many sites, ancient treponemes. All of them are post-contact. All of them are very close to this big bang when they start to diversify. And in the Americas, we also have genomes, also lineages, but they are not presented in Europe, but they are only found in the Americas. So there seems to be quite some diversity there in the Americas. And the European part is not part of that diversity. So this kind of supports the idea that this comes from the New World. And soon enough, I hope we will also have genomes from pre-contact America, and that will probably settle the question forever.
I agree. That's very cool. The next question is it's about Mycobacterial tuberculosis. So if it evolved from the mycobacteria from the seals, and also I would add that there is also mycobacteria in Europe that doesn't seem to be related to the one of the plague, no?
I mean, okay, so tuberculosis, of course, causes tuberculosis, not plague. So Mycobacterium tuberculosis has a certain diversity. There are about 7 main lineages in the world. And then the 8th lineage, or the 6th lineage, you could call it, maybe has some lineage that splits from it, which is actually all the animal lineages. And it's bizarre. It's like in the past, people thought that we got tuberculosis from cows because cows have Mycobacterium bovis. But then in the genomic age, people could actually show Mycobacterium bovis, which is the tuberculosis from cows, actually falls in the genetic diversity of humans on that one branch in one of the sub-branches of humans, where all the animals fall, including badgers, seals, lions, buffaloes, mongoose, like all kind of animals. And they all fall within Europe, like within the diversity of humans. So it seems to be actually evolved in humans and then spread to animals. So at some point, it must have spread into the seals. So I think, our hypothesis currently is that sometime, about 5,000 years ago or so, it moved into animals, from animals into seals, from seals into the southern hemisphere. In the southern hemisphere, all over Australia, New Zealand, South America, South Africa, Antarctica, you today have tuberculosis in all the marine mammals down there. So it was introduced. The genetic diversity of the strains we have in Australia, New Zealand, the Americas today are only 500 years old. So it's a recent thing that happened in the seals. It could have happened even before. We think it happened even before. But at some point, it then also made its way into the Americas. And we have a common ancestor of the lineage we found in those Peruvian and what was called Mycobacterium pinnipedii, which is basically the one we found in seals today, about 500 AD or so. So at some point, probably 1,500 years ago or so, it was present in seals or some sort of marine mammal of the coast in the Americas, and then kind of was introduced in the human population. But that particular strain is not present in the seals anymore. It's just that the closest relatives today are in seals. But they are only 1,500 years diverged from the one we found in those humans there. And there was clearly no contact at 1,500 years between the New World and the Old World. So it must be somehow that this was a vector or somehow brought it there. But it originated in humans. That's pretty clear. And at some point, it got into the seals. So this is quite a few parts of the story we haven't yet uncovered. We would have to study a lot of ancient seals, but they're usually in the stomach of a white shark or on the bottom of the sea. But we will hardly find those remains, but maybe in the future.
That's very interesting. And the next question is about pox viruses. Do you have anything to say about pox viruses?
So again, can't talk too much about unpublished data, but the published data currently has an origin of smallpox in the Old World about 500 to 600 AD. That's the common ancestor of smallpox. It might have infected the Roman Empire already. There is the Antonine plague, which some people think was smallpox. This is like 3rd century. Doesn't really fit the data too well, but there's not so much data from that time. So the oldest genomes currently are from Vikings, from about 800, 900 AD. And then from a mummy from 1700 or so. The Vikings actually had a lineage that is extinct today, which is interesting in itself. So there was some sort of Viking lineage. So there was a certain amount of genetic diversity back in the day. And we hope now with more ancient DNA data, we can actually solve that. But we're pretty certain that it was introduced by the Europeans to the New World. So it wasn't present there before. And there will be some ancient genomes from the Americas also soon. So I think then we get a little bit more of that story.
I would say a Viking smallpox looks like a very bad disease to have. The next question is how reliable it is to tell skin color from genetic data.
It depends where you are in the world. I think in Brazil, it's not reliable at all because you have so much genetic diversity, especially from Africa, that it will be very difficult. And people have actually shown that in South Africa, where you also have a lot of genetic diversity because you have people of very deep human ancestry, Khoisan are the deepest diverged lineage, and you have also East Asian ancestry and European ancestry, it doesn't work well. So basically, if you have a certain gene, it doesn't correlate so well with the actual observed skin color, which you usually kind of look at under the armpit. Because there's so many variants. I mean, there's more than 100 genes and variants that are known to influence skin color. So it's not like, you have that gene for dark, which you also see if you go to a drugstore, right? If you ever go to a drugstore, you look at the shades of makeup, right? There is not black and white. There is like everything in between, right? There is like 1,000 shades, because there's 1,000 shades of skin colors. For me, I always find it like people talk about black and white. It's like that's the two colors that don't exist, right? You don't have black people, don't have white people, but have everything in between. You really have a lot of shades, and it's just really funny. And it's also a gradient, right? There's not one or the other, and that also shows you that there's a lot of genes involved. However, in Europe, the genetic diversity is a bit limited, and there's a very good correlation between the kind of main variants that I showed, those two genes, and skin color. That works in Europe quite well. But then again, if you have people from other parts of the world, it would work differently. It doesn't work so well, but here it does work quite well. So I think we can say that. What we can probably not say is how dark was the skin of the hunter-gatherers of Europe before agriculture came here. Was it like people in Nigeria today, or was it like people in Morocco, or is it like people in Ethiopia or something like that? That's a bit harder to say. So that's, has also caused a lot of critique, because many of us in the field of archaeogenetics published this data that hunter-gatherers had dark skin, and then some reconstruction had them really dark, and then there was a TV documentary in Sweden about the first Swedish people with like people that basically looked like they came from Nigeria. And then, of course, all the right-wing parties were like, this is kind of, Vogue, and kind of like, lots of critique on those people. But I mean, to the best of our knowledge, this is how the people look like. But, we don't know exactly. But certainly they were dark. But a lot of people don't want to hear that. I mean, that really kind of hurts a lot of kind of pride nationalists in this part of the world, that people were dark-skinned a few thousand years ago.
Okay, we have 2 more questions, and then we promise to let you go. So I'm actually going to read the other one. I think the other one's more from the same person. Yeah. So the idea here is that humans, the light skin, the development of light skin in humans is actually coming together with vitamin D production. It's related to agriculture somehow. And how would this fit with the idea of like agriculture as in Africa? And how do you think this would relate? Like, do you think the light skin in humans is something how do you think this would be related to any kind of environmental patterns?
Yeah, so it seems to be very clear from our genetic data right now that the skin color changed with the introduction of agriculture. And that is a direct response to doing agriculture in the north. I mean, the amount of sunlight you have in this part of the world is just very, very small in the winter, especially if you move to the Great Britain or Sweden or something like that. So you cannot really produce a lot of vitamin D in the skin, especially if you're dark skinned. People today, in fact, especially people of South Asian or African descent, they have vitamin D deficiency living in Scotland or Ireland or Sweden or Norway. Which today is not a problem. You just get supplements in the pharmacy, and those people know that. It's also the same in North America, where you even put vitamin D in the milk. But in of course ancient times, that caused vitamin D deficiency, which is really bad. So the best way to adapt, if you don't have it in the diet, which people didn't have because early farmers were basically vegetarians. They didn't have meat and fish based on the isotope studies. They didn't eat a lot of meat and fish, so they didn't have vitamin D in the diet. So the best way to adapt then is to change skin color, basically, get white skin, because that's a direct adaptation to the living life. Hunter-gatherers didn't need that. And still today, if you look at Eskimo, they live for 4,000 years in Greenland, which is even further north, but their phenotype is actually not European. They don't look like Swedish people. They're not transparent in their skin. They're basically still dark hair, dark eyes, dark skin, because they eat a lot of fish and meat. So they don't have vitamin D deficiency. But Swedish people, 8,000 years ago, would have had it. So they had to become white. And that's why they are, I think, the whitest people, if I can say that, in the world today, because they're by far the most northern people in the world to do agriculture. There's no other population that lives so far up than Finland and Sweden that does agriculture. If it's the same latitude in North America, you're north of the Hudson Bay. You're like Fairbanks, Alaska. This is all permafrost. No one could have done agriculture anywhere there. It's just Europe has the special condition of the Gulf Stream, which pumps the warm water here. And this is why you can have agriculture in Scandinavia. This is why only Europeans became white, because we just have very different climate here, further up north than, say, in Siberia or North America. In South America, and Africa, and Australia, you're far away from that zone, right? the South Pole is so far away. I mean, well, maybe in Patagonia or something like that. But trying agriculture in Patagonia, I think even today is pretty difficult. So I think it's just basically too cold down there, and that kind of what made those Northern Europeans really white, because they have been so far up north, but doing agriculture. At least that's the best explanation. I think that fits quite well.
Yeah, that's very interesting. So the last one is about naturally ancient DNA using a combination of modern technologies of DNA sequencing, but also you have special tricks for the ancient DNA. So what's the secret? How do you get DNA from the 100,000 years old DNA?
I mean, the secret is actually, it's a lot of methods. And, Svante has been here, for many years developing all kind of molecular biology protocols to extract DNA from bones, extract DNA from soil, extract DNA, from hair, like for Ludwig von Beethoven. I mean, that's something we did, but also other people in the field obviously did development. But then also high throughput sequencing, this new technology that we applied very early on, was perfect for ancient DNA, because what it actually produces very short DNA sequences, especially at the beginning. Now they're 150 bases, but wow, I mean, Sanger sequencing that I did 20 years ago was 500 bases. So, 150 is still very short compared to the good old days with Sanger sequencing. But ancient DNA is an average 50 base pairs long, so it's highly degraded. So it was a perfect technology for us. We were driving even the development. If you look at the protocol, the Illumina standard protocol for double-strand DNA sequencing is basically developed by ancient DNA people. So it's really that we were really driving this because this was a perfect technology. We had adapted it, even before. We were the first people in Europe to have a 454 machine in 2006. And it was just because everyone else was like, who wants to have 30 base pair fragments? And we were like, oh, that's all we have. So we don't care. And we just wanted to have more and more and more. And I think, we've always been quite cutting edge in terms of method development. I think that really is the secret, right? You really kind of apply the latest shit, if I can say that, and then just always try to be cutting edge, and that has helped a lot. And now within sediment DNA, the people like Matthias Meyer, who has been also kind of developing a lot of those methods, are now working with. I think this will be also very exciting because there's a very limited number of fossils, so especially human fossils. But there's a shitload of sediment in those caves that you can analyze. So if you they get now whole genomes from sediment, and I think that will be very exciting. So that I think also in the future, this will be quite interesting. It's still limited, I think. A lot of sites don't produce it, but if you look at a lot of samples, you will actually also find human DNA in sediment, which can be interesting to say. You have a site like Monteverde in Chile. There is no human remains, but there's a lot of sediment. So maybe with the sediment, you can actually find who was there 14,000 years ago, right? And there's all those archaeology in Brazil and other places in the Americas that's supposed to be older than 15,000 years, even though I doubt it. But need to be careful with the audience here. But maybe someone does find in the sediment then some human DNA, which is different to what is there after 15,000 years. I just have a hard time to imagine that people were in the Americas before 15,000 years, because soon modern humans come anywhere, the population explodes. Always, everywhere. There's not a single example I can think of where humans just went in and it's like, oh, this is nice here, bye-bye. It's like, you wouldn't do that, right? It's like you find a continent, of course you reproduce and you have a lot of people. And that has happened through our history always, everywhere. Wherever we stepped foot on, our population increased a lot. And that would have happened in the Americas as well. And then the genetic signature would still be there. There should be still people in the early record or now that would have this ancestry, and they're not there. So I don't think there can be people in the Americas before 15,000 years ago.
So I think we need to finish now. I'm receiving a lot of threatening messages here in the chat. So
yeah, many people saying amazing lecture. It was excellent, Dr. Johannes. We are very pleased with your conference today, the Aula Magna. Great talk. Thank you very, very much for coming here and to show such very interesting data. And congratulations for your work. And we wish you much success, much more than you have. And you are invited now to come in person to Brazil.
Exactly.
Maybe in the future, in the near future, that would be a pleasure to have you here.
Thank you very much, Johannes.
Yeah. And I'd like to thank the audience and the very nice questions they posed here, and Tomás for a nice conduction of the lecture, the Aula Magna. Mm-hmm. Thank you very much, everybody.
Thank you.
I think you'll have to move now to Portuguese, if you don't mind.
I'll just say goodbye.
Thank you
Bye-bye.
very much.
Thank you.
Goodbye.
Thank you.
Goodbye, Tomás. Thank you.
Thank you, Tomás. Bye-bye.
Nós agradecemos ao professor Krause e a todos os que participaram desse evento. Neste momento, então, teremos o sorteio de brindes, o qual só foi possível graças ao apoio da editora da UFMG e o professor Marcos Calistos e Juliana Silva França. Então, agora faremos o sorteio de alguns livros, só lembrando que o número utilizado para o sorteio foi gerado segundo a ordem de confirmação de presença e o sorteio será feito online através do site sorteador. O primeiro livro a ser sorteado será o Biotechnologies and the Human Condition, oferecido pela editora UFMG. Então, vamos ver quem foi o sorteado. E o ganhador desse livro foi a Nádia Antônio Namunawa. Parabéns pelo livro. Agora, o próximo a ser sorteado será a obra intitulada Monitoramento Participativo de Rios Urbanos, cedido pela professora Juliana Silva França e Marcos Calisto. E o número sorteado foi o 73, Gabriel Antônio Mendes de Brito. O próximo livro a ser sorteado é o Manual de Computação Evolutiva e Meta-Heurística, também disponibilizado pela editora UFMG. E o número sorteado foi o número 37, que é a Ana Carolina Santos. Parabéns, Ana. O próximo livro a ser sorteado é a obra intitulada Monitoramento Participativo dos Rios Urbanos. Só conferir aqui. É esse mesmo? Monitoramento É esse mesmo, Monitoramento Participativo dos Rios Urbanos. O número sorteado foi o 33, é a Letícia Cristina Dias Lima. Parabéns, Letícia. O próximo livro a ser sorteado é a Introdução à Identificação dos Sistemas, cedido pela editora UFMG. E o número sorteado foi o número 25, que foi o Eliseu Soares de Oliveira Rocha. Parabéns, Eliseu. E o último livro a ser sorteado é a obra intitulada Monitoramento Participativo dos Rios Urbanos, também cedido pela Juliana Silva França e Marcos Calisto. E o sorteado foi o número 111, que foi a Isabela Aparecida de Souza. Parabéns, Isabela. Assim, deixamos registrado o nosso agradecimento pelo apoio ao evento. Para compor a sessão de encerramento, convidamos então a coordenadora do Núcleo de Apoio à Pós-graduação, a professora Glória Regina Franco, e a subcoordenadora do Núcleo de Apoio à Pós-graduação, a professora Érica Cristina Jorge. E passamos então a palavra para a professora Glória Franco.
Bom, primeiro eu passo a palavra para a professora Érica Jorge, que é a atual coordenadora do Núcleo de Pós-graduação.
Eu quero agradecer, né? Foi uma palestra sensacional. Acho que a gente teve muita sorte, assim, a gente vem tendo, né, palestras muito boas de aula magna. Achei essa palestra muito boa, sensacional. Espero que todos tenham gostado. Mais uma vez, quero agradecer todo mundo que ajudou, né, em especial a professora Glória e o professor Fabrício, que ajudou no contato com o Johannes. Quero agradecer o pessoal do NAPG, mais uma vez, vocês moram no meu coração, muito obrigado pelo trabalho. E desejo um ótimo ano para todos. A gente já está na luta, né, gente? Mas desejo que a gente tenha um 2023 de muito sucesso, de muito trabalho. E é isso. Fiquem atentos aos eventos do NAPG. Esse ano a gente tem muitas novidades e contamos aí com a participação de todos. Um abraço para todos.
Professora Glória, gostaria de falar um pouquinho?
Obrigada, Amanda. Então, gostaria também de agradecer a presença de todos nesse evento. Foi uma escolha muito acertada, né, trazer o professor Juanes. Agradeço muito ao Fabrício Santos, também ao Tomás Pinotti, e imagino que vocês tenham tido assim, uma ideia do que a ciência, até onde a ciência pode ir, né, usando sequenciamentos de genomas antigos. Acho que foi maravilhoso e também a gente entender como isso foi possível ser laureado com o Prêmio Nobel, né, o professor Svante Paabo. E todo o grupo que trabalha junto é um trabalho de muitas mãos, né, assim como toda a ciência mundial. Então, eu desejo a vocês um excelente ano, fiquem atentos às nossas atividades, nós temos seminários NAPG nesse canal, que são muito bem divulgados, né, pelo site da NAPG. A gente pede sempre que os alunos, caso precisem, né, nos peçam coisas, nos enviem sugestões, é sempre bom ouvir vocês, né, e não deixem de ir nos eventos que a gente convida, tá? E divulgar também o nosso site do YouTube e fazer parte dessa família NAPG. Nós estamos procurando novos membros para a nossa equipe, todos são muito bem-vindos. É só escrever para o nosso e-mail, que é o napg@icb.ufmg.br, e a Rosemary, nossa querida secretária técnica administrativa, vai responder vocês, a gente pode conversar, tá? Estamos aí à disposição para atender a todos. Então, agradeço mais uma vez essa equipe maravilhosa que nós temos, de alunos de pós-graduação muito empenhados, e a nossa técnica administrativa, Rosemary Castro. E agradeço também a parceria com a professora Érica, maravilhosa. Uhum. Obrigada a todos, bom final de semana, né? Ainda temos um restinho de semana aí, e bom semestre.
Agradecemos a todos pela participação e fiquem atentos, então, aos próximos eventos promovidos pela NAPG, como a professora Glória falou. E assim encerramos, então, a nossa aula magna da pós-graduação do ICB de 2023. Muito obrigada.